DNA is made up of two anti-parallel strands that are wound together to form a double helix. However, this is a graphic of the double helix unraveled. Each of the strands are made up of a sugar phosphate backbone, with the red dots as the phosphate and a pentagon as the deoxyribose sugar. Each phosphate is attached to the sugar with a sugar phosphate bond, which runs from the fourth carbon on the sugar to the phosphate. And this occurs all the way down the backbone. And if you look at the strand on the right, it runs in a 5' to 3' direction, with the fifth carbon at the top and the third carbon at the bottom. Since this is an anti-parallel strand, the other strand will run in the opposite direction, with a 3' to 5' direction, with a 3' at the top and a 5' at the bottom. Now that is our sugar phosphate backbone. On the interior of the DNA are the 4 nucleotide bases. Each base has a complementary base, and between each base pair is a hydrogen bond. Now, there are both purines and pyrimidines. Purines include adenine and guanine, and pyrimidines include thymine and cytosine. Guanine and cytosine are both base pairs. They are complementary to each other. They fit together like a puzzle piece. Adenine and thymine, however, are also complementary of each other, and they are base pairs as well. Between the two are 2 hydrogen bonds. Between guanine and cytosine, there are 3 hydrogen bonds. The hydrogen bonds that hold the whole DNA strand together are strong enough to maintain this shape, but they are also weak enough to break apart during replication and transcription. DNA replication is a process through which DNA is separated into 2 single strands and then copied to form 2 identical partially new strands, hence why this process is often considered semi-conservative. The process begins with the enzyme helicase, which unwinds and separates the double-stranded DNA using ATP. It breaks the hydrogen bonds between each of the base pairs beginning at the replication fork. Then, single-stranded binding proteins bind each of the strands in order to prevent them from reannealing or being digested by the nucleases. The next enzyme used is the RNA primase. RNA primase generates a short RNA primer on each of the template strands. These primers are the initiation points for replication on each strand. The leading strand is a strand that has a 5' to 3' direction. The lagging strand has a 3' to 5' direction. The DNA polymerase III attaches to the 3' end of the leading strand at the RNA primer and replaces the primer with nucleotides. It creates the new strand in a 5' to 3' direction, in the same direction as a helicase, as it forms the complementary bases using the free nucleotides. DNA polymerase I, in contrast, will move on the lagging strand. It will move away from the replication fork and in the opposite direction of the helicase, so it will need multiple RNA primers and multiple initiation points. The polymerase will again remove the RNA primers and replace them with nucleotides and collect free-floating bases to complement the nitrogenous bases on the parent strand. It will have to start again and again on each RNA primer. Because there are multiple initiation points on the lagging strand, Okazaki fragments form and result. DNA ligase is the enzyme that then joins these fragments together to form a continuous strand. Transcription. Transcription is the process of making mRNA from a DNA template. It is this mRNA that will later be used to help make proteins. On the DNA template, the gene being coded begins with a promoter region and then ends in it with a terminator region. The promoter region of DNA acts as an initiation recognition site for the RNA polymerase to bind. The majority of gene expression is controlled after the promoter at the operator through either enhancers or inhibitors. Once the RNA polymerase binds to the promoter, this causes the DNA double helix to unwind and open. The RNA polymerase then slides along the DNA strand, creating the mRNA in a 5' to 3' direction, making complementary bases. In mRNA, the base pairs are the same as DNA, except instead of thymine, there is uracil. Therefore, uracil is a new base pair for adenine. mRNA is also single-stranded and has a ribose sugar instead of a deoxyribose sugar. When the polymerase reaches the termination region, the mRNA, DNA, and RNA polymerase all dissociate from each other. mRNA then leaves the nucleus through the nucleus pore. Translation is the synthesis of a protein from an mRNA template. Eukaryotic mRNA has a 3' end called the poly A tail. It also has codons that code for specific amino acids. There is a methylated cap found at the 5' end. Anticodons found on tRNA are complementary to the codons on mRNA to which it binds. Attached to the end of the tRNA is a corresponding amino acid. Translation begins when the small ribosomal subunit attaches to the methylated cap and moves to the translation initiation site. The first mRNA codon is typically AUG. After the first tRNA binds to the mRNA, the large ribosomal subunit attaches to form the P site and the A site. The first tRNA occupies the P site, and the second tRNA enters the A site and is complementary to the second codon. The first amino acid is transferred to the A site on top of the second amino acid, and the first tRNA exits. The ribosome moves along the mRNA, and the next tRNA enters. When a stop codon on the mRNA is encountered in the A site, a release factor enters the A site and translation is terminated. The ribosome dissociates and the newly formed polypeptide is released.